Hello students, this is Dr. Faizan Mirza. I'm covering the aspects of how the species diversity can be assessed in a region when we are considering biodiversity. So how species diversity can be assessed? First of all, you need to mark the specific area in the field where you are going, and then you need to carry out sampling. When you carry out sampling, you need to see whether the sampling can be carried out in a random sampling manner or a systematic sampling. Now, what exactly is random sampling? What systematic sampling? That's what we'll be discussing. So when you are assessing species diversity, the first thing is that you mark the area. When you carry out sampling, you go for either random sampling or systematic sampling. What is random sampling? It is carried out when the abiotic factors in that marked field are consistent. They are just the same all across. But systematic sampling is to be carried out when the abiotic factors are not consistent throughout the field. For example, if the soil moisture content is changing along the field, or for example, there's the difference in the altitude, or there's difference in the sunlight exposure, or any abiotic factor is being changed. What do you do? Once the sampling has been carried out, the next step is you collect the data. When you collect the data, you use observation skills, you refer to photographs which are available for different phenotypes of the same species, you count them. You collect and count, you use an abundance scale. You can probably just count them in numbers, or you can use abundance scales if counting is not possible. Collection and counting can be done using pooters, sweeping nets, traps, pitfalls, quadrates, mark release, and recapture. There are various methods of collecting and counting organisms depending on the size of organism, the type of organism, and for example, if it's an insect flying, you can use sweeping nets for that. If it's a land dwelling small animal, you can probably use just traps or pitfalls for it. Similarly, quadrates can be used for plants. Abundance scale is used when counting is not possible. You use a brown blanket cover scale, and you use a kite diagram for graphical representation of the data. The next thing is that you make a list, or you just catalog all of them, and you just state the numbers of each of these. You calculate species frequency, species density. You can also calculate the Simpson's index of diversity. You plot a scatter plot to see how the values of these individual counts, how are they distributed. You draw a scatter plot. You calculate a correlation coefficient, which can be calculated by the Spearman rank correlation or Pearson linear correlation. We'll be discussing them later, how these two statistical tests are carried out. This individual here, you can see he's actually hanging with this harness somewhere, and he is just inserted this tube into this region at the pot, or at the region under the plant, so that he can collect these insects, the tiny insects, and they are being collected using pooter. This device is called a pooter. This tube, he just sucks it using his the pressure from his mouth, which is generated from his chest cavity. He pulls the air here. The air here, when he's pulling, there's a you can say a diaphragm here that doesn't allow the insects to go into his mouth. The insects are collected from this tube into the narrow tubing, and eventually into this glass test tube, and then they can be counted. So at the description of all these instruments which are being used, they will be given to you. Coordinate is something, mark, release, and recapture, abundance scale are the things that you need to cram and you need to keep in mind. How are they used? It's worth recalling the definitions of population, niche, species, ecosystem, and habitat. Now, how do you carry out random sampling? Random sampling can be carried out. How do you do that? You just mark the field with the measure tape, and then you measure its length and width, and you feed the length and width area into a random number generator. What is a random number generator? It's an app in the phone or a function in a scientific calculator. And then you ask the app to generate a set of 10 coordinates of the field, and you use a quadrate to just go and place these specific sized quadrates, these frameworks, at a specific site within the field, and you just start to catalog and count the species of that particular plant which are present in each quadrate. You may use brown and blanket cover scale if counting is not possible, which means if, for example, if it's grasses, or it's too many of organisms there which are growing together. You calculate species frequency, species density. So you can see in this photograph that this is the measure tape that was laid down. The length and width were fed into the random number generator app, and it gave the coordinate here. So the team members who would have gone for this study, they would have walked on x-axis and y-axis according to the coordinate that were generated with the random number generator app. For example, if they had received 600 meter by 100 meters, so they would go 100 on one axis and 600 on the other, and they would just place a coordinate here. This coordinate is 1.06 meters squared, this framework. So they just place a framework here. And this frame, when it's laid down, inside this frame there are plants. The group will then start to just count the cat. They initially will just catalog which type of species are here, and then they will start to count how many of each are present. With this one coordinate done, another set of coordinate will be generated using the random number generator app, and the coordinate will then be placed in that second position. Similarly, you can just go on and place the coordinate at 10 different sites within the marked area. So that will end up, you will end up accounting for 10 different positions within the marked area. And since the positions where you were counting, you are not choosing them yourself. You could have added bias to it. You probably could have not placed the coordinate in a barren area, or you probably would have placed it deliberately in a place where the plants are highly diverse, or many of them are growing together. But when you use a random number generator app, it avoids these bias. When the bias is avoided, whatever coordinate it gives you, you have to go and just place the coordinate there. There's no other option about it. So it helps in avoiding bias. We use a species frequency and a species density. Species frequency is a measure of a chance of a particular species being found in any one of the coordinate. How do you calculate a species frequency? The total number of coordinates having the organism is divided by total number of coordinates, you multiply it by 100. How is the species density calculated? A species density takes into consideration the total area counted. So we'll be using the number of individuals of a certain organism per unit area. So number of individuals counted divided by total area counted. For example, this question here, what does this question tell you? The question mentions to you that a survey was carried out on a rocky shore in New Zealand. A coordinate of 0.25 meters squared was placed. You can see it was placed 10 times. Among these 10 times, the red sea anemone was found in these 5 of these coordinates. So 1, 2, 3, 4, 5. So what's the frequency of you coming across this red sea anemone? The species frequency is 5 divided by 10. 5 means the number of coordinates in which the organism was present, divided by the total number of coordinates that you place. So that's 5 by 10 multiplied by 100. You get a 50% answer here. When you are calculating a species density, you have to take into consideration the total area covered, or the total area that you have counted. Since the area of one of the coordinates is 0.25 meters squared, and you ended up placing the coordinate 10 times, so the total area that you have counted is 0.25 meters squared multiplied by 10. So that will become 2.5 meters squared. So you have actually went through 2.5 meters squared of area, trying to figure out how many organisms are there. So how will you calculate the species density? You just add up all the numbers where this red sea anemone was present. For example, 3 plus 1 plus 5 plus 2 plus 1. You add these up, it comes out to be 12. You divide by 2.5, you get 4.8 per meter squared. What does the value of 4.8 per meter squared represent? It represents that red sea anemone is present almost 4.8 in every meter. The same example can be seen here as well. We have counted day flowers on lawn and day flowers in field, placing the coordinate 10 times. The size of the coordinate was 1.0 meters squared. It means that the total area counted was 10 meters squared, because we'll multiply it by 10 because the coordinate was placed 10 times. A species frequency in day flower on lawn and field can be compared. For example, if the day flower on lawn is counted, so it's present in 1, 2, 3, 4, 5, and 6 of the coordinates. So it's 6 by 10. 10 is the total number of coordinates that were used, multiplied by 100. So 60% is the species frequency. Whereas in the field, it's present in 1, 2, and 3 of the 10 coordinates. So 3 by 10 multiplied by 100, which turns out to be 30%. So you can see, where is the species more frequent? It's more frequent in the lawn because it's 60% there, where it's just 30% on the day field. In the field, the day flowers in the field. How will you calculate the species density? You'll just add the values and you'll divide them by the total area. You will get it for the lawn. You will add the total values and you'll just divide by the total area. You get it for the field. So it comes out to be 1.7 per meter squared or 0.8 per meter squared accordingly. You can use abundance scale when counting is not possible, or when the grasses are growing with interconnected roots, or for example when there are mosses. too many of things to count. So what you do, you'll just go and estimate the percentage cover using an abundance scale. How do you do that? You'll use this brown blanket cover scale. The brown blanket cover scale, you make use of a quadrate, but this quadrate in turn is divided into 100 small squares. So wherever you place this quadrate, you will just see in how many squares of 100 is this organism present. So this is exactly what we did. You can say these team members, they have marked this area with this measure tape. They have placed this quadrate. This quadrate is of a certain size, and it's having 100 small squares. So if they are counting a particular species, for example, if this species is being counted, which I'm just drawing with these yellow dots, so they see how many squares is it present in. And let's suppose if they just are able to decipher that this is present somewhere between 6 and 25 squares, or 6 and 25% of this cover, this entire box, so they will give it a score of 2. This is the brown blanket cover scale. So when the percentage cover is between 6 and 25, you give it 2. When there are very few plants covering less than 1%, so its value is just plus. If the cover is 1 to 5%, means 1 to 5 squares, these small squares are having the organism, you will give the value of 1, and so on and so forth. Similarly, for example, if this red thing, this red organism here, this is just present here, so this is between 1 and 5%, so this is not more than that. So yeah, the score in this case will be 1. And for example, if this blue organism here, and the blue one that I've just marked as a dot, so this happens to be there only in one of the squares, so the score will be plus. So that's how you just use a brown blanket cover scale to estimate the percentage cover when counting is not possible. That's it from my side. Thank you so much.